Ladies and gentlemen, good afternoon. Thank you for attending our presentation. My name is Tom Durig, and I am the Global Head of Pharmaceutical R&D at Ashland. The title of today's presentation is Reducing Formulation Risk Through Science-Based Excipient Selection. But on a little further reflection, the title should probably actually read Bringing Well-Formulated, Safer Oral Solid Dosage Forms to Market Faster by Replacing Empirical Excipient Selection with a Science-Based Approach, making your boss, your shareholders, possibly your family and customers happier, and eventually also you. To address this topic this afternoon, what we'd like to do is take a look at some of the drivers in the industry that have brought us to this point. Then take a quick look at the critical role and function of excipients in dosage form development, and then also take a look at why the understanding of structure-function property relationships of excipients is critical. And then we'll close with a series of case studies applying these approaches to modified release dosage form development, wet binder granulation selection, reducing alcohol-induced dose dumping in modified release dosage forms, and improving bioavailability and solubilization of poorly soluble APIs. But first, let's take a look at some of the drivers which have brought us to this stage in the industry. Essentially, there have been 2 decades of unprecedented challenges that have been confronting the pharmaceutical industry, which has led to significant structural change, as well as introduction of significant risk mitigation strategies, which have changed how the industry approaches development nowadays. Some of these challenges would be things like the pressure on revenues and stock price performance, the reduced NCE approval rates, as well as inadequately deep late-stage pipelines, and with all the low-hanging, easier fruit exhausted, more challenging therapeutic targets. And this has led the industry to respond in a number of ways, which includes, among others, the externalization of discovery and R&D assets, the increase of forming risk-sharing partnerships for drug development, a shift in therapeutic area focus towards areas such as cancer, diabetes, CNS, as well as rare diseases. And in terms of formulation development, a move towards more science-based formulation approaches, which among others would include the emergence of the quality by design regulatory as well as development framework, the increased use of computational and in silico formulation development methods, where one essentially attempts to predict a formulation before you actually go in the lab and do the work, and also more rational excipient selection approaches based on structure-function relationships. Before we talk about structure-function relationships in excipients, it's important, if one thinks about science-based formulation development, to realize and acknowledge the critical importance that excipients play in a pharmaceutical formulation. Traditionally, perhaps, excipients were regarded as the inactive materials and somewhat peripheral to the formulation as a whole, with more attention focused on developing the API properties and understanding exactly how the API affects the formulation. But in effect, whether it's by design or accidentally, excipients play a critical function in terms of ensuring that the final dosage form is able to be manufactured in a robust manner. They play a critical function to ensure bioavailability as well as stability of the drug. Therefore, excipients are one of the key factors that affect the development of a robust, safe, and efficacious drug product. When one then thinks about making rational choices around excipients, it's really important to understand structure-function relationships. Essentially, structure-function or structure-property relationships would be defined as understanding how the chemical or molecular structure and the physical or morphological structure of an excipient affects its physical and chemical properties, that affects its end-use functionality. And this is directly related to quality by design, or QBD, which today is the regulatory framework for developing formulations in the United States. QBD is defined as a systematic and risk-based and science-based and holistic approach to pharmaceutical development. And within QBD, within the QBD framework, there is a requirement to identify critical material attributes, or CMAs in short. And these are defined as physical, chemical, biological, or microbiological properties and characteristics of an input material that should be within an appropriate limit to ensure a desired quality. So in short, in order to identify what those critical material attributes would be and what the appropriate limit would be, quantitative measurement would be, it's really key and critical to understand the structure-property relationship of an excipient. To illustrate this in a little bit more detail, structure-property and structure-function relationships can more or less be divided into two groups, as you see here on this slide here. This first group on the right would be fundamental structural properties, which are due to the chemical structure or molecular structure. So characteristics such as perhaps molecular weight distribution, substituents, if it's a polymeric excipient, the degree of substitution on the polymer, or the presence of certain functional groups in the excipient. And these generally lead to secondary material properties, such as, for instance, the elastic modulus of a material, or the glass transition temperature or melting point, which in turn imparts specific types of functionality or in terms of its end use. For instance, the tablet strength or tablet hardness could be impacted by these secondary material properties that go all the way back to the chemical structure of the excipient. Another example would be film coating strength and perhaps the incidence or the defect rate in a film coating during manufacturing. The second group of structure function properties would be these here, which we tend to think of as being more imparted onto the excipient due to the production process. They tend to be more physical properties, things perhaps like particle or morphological properties. So particle size distribution, density of maybe bulk density of an excipient, as well as particle shape. And then these would lead to secondary material properties, such, for instance, bulk powder flow, which in turn, as we well know, can affect the final content uniformity of a tablet, ensuring that the patient gets the right dose every time. To illustrate these concepts in a little bit more detail then, let's take a look at some of our case studies. And this first case study is an example of using a science-based polymer design approach to eliminate modified release profile variability in a hydrophilic matrix tablet. And the hydrophilic matrix tablet here in this case is one that uses HPMC as the controlled release polymer. As we all well know, HPMC is the most widely used modified release polymer for this kind of application, and yet only, if you look at the traditional situation, yet only a limited number of molecular weight grades have been commercially available on the market for this kind of application when we started this work. And this immediately, highlights a problem. Here we have an example of glipizide, which is a moderately soluble drug, and incorporating 25% of all these different polymer grades that were commercially available at the time. You can immediately see that there's a lot of white space here in the 24-hour factor space. And the 24-hour duration for drug release is the physiologically relevant one in the case of oral drug delivery. And as you can see here, if, for instance, a formulator needed to achieve a release rate that fell somewhere in between here in this white space, they really wouldn't have an existing product that they could choose. Most likely, the option would be to maybe do the logical thing and try a blend of the blue one here, which happens to be 100 centipoise HPMC, and blend that with this red one here, which is Benecel HPMC K4M, 4000 CPS. And maybe we would achieve an intermediate viscosity grade, giving us an intermediate release. But there's a problem with that, a significant problem, because although we can essentially blend a 100 centipoise HPMC with a 4,000 centipoise HPMC to make an intermediate 750 centipoise HPMC blend, what we're in effect doing is, as you can see here in the bottom left corner, we're blending essentially short polymer chains and long polymer chains to arrive at some intermediate viscosity, which as a result has a very wide bimodal and a very wide molecular weight distribution. This is very different from if we actually custom made an HPMC polymer that has exactly 750 centipoise as its viscosity, achieving that through an intermediate polymer chain length as shown here in the top left corner. And this is, physically obviously quite different. And this here we would have a more uniform and narrow molecular weight distribution. Molecular weight tends to be measured as polydispersity index, which is the average of the weight average and number average molecular weight. And typically, in most polymer scientists' book of rule of thumbs, we would say, a polydispersity index of less than 5 is usually regarded as relatively narrow and not too wide. And you can see here clearly that if we use this polymer blend where we have the bimodal distribution, we get a polydispersity index of about 8, contrasting that with the custom-made HPMC of 750 centipoise, half the polydispersity index. And this is very, very significant because if we actually then look at the properties of this gel-forming tablet, and here we have the gel strength for the HPMC tablet containing the blend, that's the red line, and then the one where we have the custom-made HPMC with 750 centipoise, we see a significant difference in gel strength of these two, which is due to the fact that we have a different physical situation. And this has direct effect, this directly impacts the release profiles, as you can see here, because the gel strength impacts the erosion as well as the variability of the erosion of the tablets. And clearly you can see here that the custom-made Benecel K750, the one with the unimodal molecular weight distribution, has a far narrower variability than this very variable blend of the two polymers. Another example of using a science-based polymer selection approach would be the choice of tablet binders for wet granulation. Here we have a simplified schematic of the mechanism by which granules are formed during wet granulation, and it starts by the binder droplets contacting a powder bed, and if the binder solution is spreadable and able to wet the API and the formulation contents, then one tends to form a smaller nucleus, which then grows into a larger granule. And if this binder solution, on the other hand, has relatively poor wettability, one tends to get this scenario here where the contact angle is very large. The binder solution is really not able to spread very well over the powder bed as such. resulting usually in more inhomogeneous and poor binder distribution, weaker and more porous granules. On the other hand, if we have good wetting, if the binder solution is chosen such that it has good wetting of the API, we have this situation here where the contact angle is close to 0, and you have good contact between the binder solution and the API, resulting in homogeneous and denser particle formation or granule formation, resulting in strong granules. This ability to spread for the binder to wet and spread over the powder bed can be measured as the spreading coefficient, which here you can see in this equation is the function of surface tension as well as the contact angle. So it's surface tension of the binder solution and the contact angle on the API surface. Using this approach, our scientists have conducted some studies where we systematically varied the surface tension of the binder solution by choosing different binders that give different surface tensions, and looking at this for different hydrophobicity APIs, basically drug molecules with different hydrophobicity. In this case, we measured the hydrophobicity as the log P. And as you can clearly see here, for a low log P, so very hydrophilic molecule such as metformin, clearly the PVP and copovidone, which have the higher surface tensions due to their more hydrophilic nature, give much stronger tablets in terms of tablet compactability as compared to the lower surface tension of HPC and HPMC. As we move up this hydrophobicity scale, we can see perhaps for albendazole, the red series, it really doesn't matter much in this intermediate hydrophobicity range. All the binders perform similarly, but then as we move up the chain here towards the more hydrophobic APIs such as efavirenz and simvastatin, clearly the HPC stands out as giving far more compactable tablets than the other tablet binders. So this is an approach that can be used to basically make quantitative and qualitative predictions of which tablet binder might work better based on some simple measurements of surface tension and knowing the log P of the API, which then allows it to make it possible to set up a science-based wet binder selection decision tree, starting with the question, what is the log P, what is the hydrophobicity and wettability of my API? In the case of a low log P and a very hydrophilic API, maybe less than 1, plasdone, PVP, or copovidone would clearly be the ones we would recommend. If there is a medium hydrophobicity in this range here of log P 1 to 3.5, either plasdone, PVP, or Klucel HPC would work equally well. And for a very high log P API, we would typically recommend Klucel HPC. The next case study is one where I'd like to look at reducing alcohol-induced dose dumping in modified release. This is an area that's attracted increasing attention over the last 10 years or so. A triggering case for the FDA was in 2004 with the introduction of a new hydromorphone extended-release capsule. This capsule contained extended-release beads, which comprised alcohol-soluble excipients like methacrylate, copolymer, and ethyl cellulose and stearic acid, which are commonly used as controlled-release film coatings because they're relatively water-insoluble. But in this case, were unfortunately alcohol-soluble. And the issue here was that upon ingestion of large quantities of alcohol together with this particular dosage form, there was the potential for the controlled release mechanism to be essentially obviated. And as you can see here, as is illustrated in this particular resolution profile, based on studies from FDA at the time, the typical release profile is shown here. Desired controlled release profile is the blue line. And then as one increases the alcohol content in the dissolution medium, up to 40%, which simulates a relatively extreme case of very large amount of alcohol ingestion, we can see that the dosage form moves from being a modified release, controlled release one, to essentially an immediate release one. And the combination of a large amount of alcohol and this very potent opioid narcotic has the effect of depressing CNS function, causing a respiratory depression and even death. Here again, there is an opportunity to approach this through a science-based approach in terms of excipient selection, because in contrast to ethyl cellulose and methacrylate copolymer, which are extremely hydrophobic and not soluble in alcohol at all, not soluble in water at all, pardon me, if we look at some of the other modified release polymers, controlled release polymers, such as HPMC and HPC, which have a better balance between hydrophobic and hydrophilic properties, we can actually see that in this case, these polymers not only form gels, but even slightly increase the viscosity as one adds ethanol to an aqueous solution. So in fact, up to 40% ethanol in an aqueous solution, these are very robust gel-forming polymers. And so therefore, not surprisingly, when making dosage forms with these matrix polymers, such as HPMC or even HEC as well as HPC, there's really no effect in terms of dose dumping as we increase the alcohol content of the dissolution medium from 0 to 40%. In fact, there's a slight retardation in the release profile as we add more alcohol due to the increased gel strength. And as you can see here, if you look at the matrix structure of the tablets after dissolution, it's quite clear, 5 hours dissolution, that the matrix structure looks very similar, whether it's simulated gastric fluid or simulated gastric fluid with 40% alcohol. As a last example then, for this afternoon, I'd like to just quickly illustrate how our scientists are using science-based approaches to redesign the molecular structure of HPMCAS to develop more efficient drug delivery polymers. In this case, what we're attempting to do is to improve the drug-polymer interaction in an amorphous dispersion environment so that we can achieve enhanced solubilization and supersaturation for some of the APIs for which today there are really not many feasible answers. And so if we look at the schematic here, this would be the classical HPMCAS, which was initially developed more for capsules and as an enteric coating, and rather accidentally found use as a solubilization polymer. And by using new synthesis methods, our scientists have found a way to rearrange these acetyl and succinyl side groups on the HPMCAS polymer to allow more efficient interaction with drug molecules. And these redesigned HPMCAS analogs, are enabling the delivery of more difficult drugs, for which currently there's really not too many options to solubilize them. And by way of example, here we have an example of solubilization of a model drug, which is ezetimibe, which is used here as a non-proprietary model of a rapidly recrystallizing, rapidly precipitating polymer. And as you can see, with our new HPMCAS analogs over native ezetimibe, we're achieving something like a 7-fold increase in solubility, nonetheless at a 60% drug load. And we also are achieving a significant improvement over our current HPMCAS, which shows a very rapid precipitation after initial solubilization. Then the last example here is for very highly hydrophobic drugs. There are drugs which are extremely hydrophobic, very high log Ps. In this case, we're using Probucol as a non-proprietary example of a high log P compound that has a log P of 10. And as you can see here again, our new analogs of HPMC are able to achieve significant improvement in terms of solubilization over the currently available HPMCAS. This then brings me to the end of our presentation this afternoon. I hope I have been able to convince you that science-based excipient selection is an effective and widely applicable means to enhance formulation quality and reduce formulation development risks. Thank you very much. Be happy to take a few questions before the next talk. Thank you.